This afternoon is being co-hosted by Mediterranean Gardening Margaret River, who are providing the delicious afternoon tea for you, and U3A, who, as always, have wonderful volunteer tech people. Thank you, Greg, who can produce this. So we are also videoing today, videoing John, not you, so that we can share this with our affiliated garden groups. I might add around the world. They've shown so much interest in this part of our program this year that I hope nobody has any objection, but I don't think Greg's aiming to get any faces other than John's in here. So it gives me so much pleasure to introduce you today to our illustrious speaker, Dr. John Huisman, Curator of the Herbarium since 1985. And looking at John, who I hadn't met before, I think he must have been a child when he started. When I first approached the herbarium, I was imagining that enticing someone like John to come down here was a little presumptuous. But I forgot about the lure of our fabulous marine life. And I know John's aiming to hit the water while he's down here. So let's hope we can find you something interesting, John, while you're in the region. John's CV is so impressive and lengthy, I'm not even going to try and cover it. However, his labour of love is his interest and study of our amazing plant life and underwater photography of the same. In his own words, I am a phycologist. Now, I did have to look that up, John. Primarily interested in the taxonomy of Western Australian seaweeds. My goal in life is to document photographically and imprint the entire marine flora of WA. I also hope to alter the perception of seaweeds as smelly beach drift. They include many spectacular and attractive plants, broadly speaking, and deserve a better image. There will be a short opportunity at the end of John's talk for questions, and John has a few of his latest books here for sale. Could you all please give a warm welcome to Dr. John Huisman.
Thank you very much, Julie, and it's lovely to see such a full house. I must admit I wasn't expecting so many people for a talk about seaweeds. I'm hoping it's a reflection of me as well as the seaweeds. So people can hear me okay, no problem, and you can see the slides okay. So Julie, I do have to correct you a little bit in that I wasn't the curator in 1985. I did my PhD in 1985 and then spent many short contracts doing research projects. Actually spent a couple of years in Hawaii doing seaweed taxonomy, hence the shirt. It's changed my attire for the rest of my life. And in 2016, I managed to get the position of the Curator of the Western Australian Herbarium, which is an odd position. It's an odd title for the position. It's actually the Director of the Herbarium. The curators of most institutions are actually responsible for a small collection or a particular collection. I'm responsible for the whole thing, which is quite weird because I'm not actually a trained botanist. And the herbarium is Western Australia's official collection of dried plant specimens. I'm explaining this because somebody has asked me before. Whenever I give tours of the herbarium, so if you're ever in Perth, do come and do a tour. It's the official collection of dried plant specimens. We have about 890,000 of them at the moment. The herbarium has actually been in existence since 1929, so we're coming up to our centenary. It's going to be a big celebration. And we've got specimens that were collected from some of the earliest explorations of Western Australia. I've actually got a specimen, and I put this out for the tour always. Specimen was collected by Banks and Solander on Cook's voyage in 1770, and it's still in perfect condition. So it's a big responsibility to maintain this collection because there are things called herbarium beetles, and if they get into your collection, they can absolutely decimate it. So we're very strict on what we let in and how we let things in. Now I'm really going to be just talking about seaweeds today, which is my interest. As I said, it was 1985, they actually had seaweeds in multiple projects. But as Joy said, my aim for many, many years is to document it. And I think Julie mentioned before about how wonderful it is to have a job that is your passion as well, which is absolutely true. I get to go out and take photos of seaweeds and collect seaweeds and press them, and it's part of my work. And producing books like this. Please do. I know I have to put in a sales pitch. Please do come and have a look at the book and see most of my colour photos. Now we'll see if this works, Greg. Oh, just as an aside. This is one of the beautiful seaweeds. What's that? No, we're going to go back now. Sorry, I pressed the wrong button. No, there we go. This is actually a seaweed called Griffithia, and these individual cells here, which are probably about the size of a thumbnail, so they're quite large for individual cells. And these little reproductive structures here, so they're like little sort of protective branches here, and these spores, spore packets here. And it's just one of many, many seaweeds that for me, they're just visually impressive. And I go to Point Perrin quite regularly to take photos, and people tell me, haven't you collected and photographed everything there? I said, there's always something new, and you can always get a better photo. That's the other thing. Okay, so the history part of what I'm going to talk about today, I'm only going to concentrate on a guy called William Henry Harvey, who was an Irish botanist who came to WA in 1854 and collected a whole bunch of seaweeds. I'm not going to talk about indigenous use of seaweeds because really there's not much recorded about indigenous use of seaweeds. Some of the earlier collections were actually made on the voyages of discovery, so that when the French and the English came to Western Australia, they collected seaweeds and they took them back to the UK or to Paris and described some of these. And so even there were seaweeds collected on the Recherche and Esperance expeditions, things called the bull kelp, the Villia potatorum, which we don't have here. It's quite an amazing seaweed. It's one of the few that's actually there's an industry associated with the Villia. I said bull kelp is called, and it actually grows in a shallow intertidal and just subtidal, and on really rough coasts, incredibly rough coasts around Tasmania and Victoria. And this holdfast here, you can often find this stuff washed up on the beach, and that holdfast is so strong, it's usually the rock that breaks rather than the holdfast. And so they use this one. This thing is you can harvest it. They harvest it for alginates, but they're only allowed to take feed strip. You're not allowed to actually take it from the shore, growing from the shore. It's very, very impressive seaweed. This is pretty much where it grows. These maps here, one of the things that the herbarium does, and every herbarium around Australia, is that we put our information, species information, into a thing called the Australasian Virtual Herbarium. And you can get on the web and go, okay, where does a particular species grow? And it will bring up dots on maps that show you where things are. And obviously they're not complete because these things are actually collections that are held in herbarium. So it's not necessarily the full distribution, but it does give you an idea of where you can find these sorts of things. But this is one of the ones that was collected and described by the earlier French expedition. It's one of the few that actually does have a very strong indigenous use, recognised use, and it's actually called potatorum because of this, because the Tasmanian Aboriginals used to use it for a water carrier. And so they would fashion it into, this is the kelp here, fashion it into a container for carrying water, and that's the potatorum reflects that. So. Okay, just some of the other things. There's a thing called Acetabularia colliculus. Amazing looking thing. These are all these individual things are just one cell, and they've got sort of calcified stalk, these wine cups that grow at the tips there. I'm always amazed by the diversity of things. People see with books on mushrooms and things like that, incredible shapes and things like that. And seaweeds are the equivalent, I would say, easily. So coming to WA, there's some seaweeds collected down at King George Sound. Robert Brown was the botanist who described them, and one of the ones that he described was a thing called Hormosira banksii, which some of you might see. We don't actually get it around the west coast of WA. It probably is as far west as Denmark maybe, intertidal seaweed. But if you're in Victoria, it's absolutely, it blankets the reefs, the intertidal reefs, called Ormosaurophaxii. That just gives you, again, this idea of where you can find it. So one of the things about being a taxonomist is that, when I say people have collected these things and describe species, one of the rules if you describe a species is you actually have to have a specimen yet. You can't just say, I'm going to call this something, and then that's it. You have to actually have a specimen lodged in a herbarium. Otherwise, the description of that species doesn't count. And so a lot of these species are actually, as I said, sitting in overseas herbarium. This is one that's in the British Museum, Natural History Museum of Britain. And it doesn't look like much. You can see a few shells here and things like that, but it's certainly what was collected by Brown, or described by Brown from Australia in 1800. And then he produced these illustrations of it. It was called Fucus paniculus at the time. And it was quite weird because there are so many genera of seaweeds now, but at that time there were 4. Everything was either fucus, where it was big enough, or conferva, if it was hairy, or ulva, if it was foliose, or cara. Cara was the other one, it was actually quite distinctive. This is what it looks like growing up at Shark Bay. So this is another one of the ones that's been described, and this is the only kelp that grows in Western Australia. It's been called a Clonia radiata. Kelps are very unusual in that they actually grow from this point here. So this is the actively growing area here, and at the base of these branches as well. But so at the tips there, that's the oldest part of the plant. Whereas normally you would think the tip was the young part, but they actually grow from this part here. And the kelps, like Macrocystis, which form those big kelp forests in Tasmania, do exactly the same thing. And they have an alternate life cycle. So these large kelps actually have a tiny little microscopic stage. It's the gametophyte, the sexual stage. This is actually the sporophyte stage. So it's a complicated life history. Okay, so one of the resident collectors was a guy called Johann August Ludwig Price, and you probably recognise Price's name. There are quite a few species named for him, and he collected a whole bunch of stuff. I put this photo, this image in there as a lesson to me more than anybody else, because there are no images of Price available. No one's ever had an image. And I had a friend and I said, "Can you reckon you could find one?" And so she went on the internet. And of course, the internet is such a font of, just, you can accept anything, Anyway, so this photo came up, and it was associated with a biography of Price, so, it seemed legitimate. And we all got very excited. We've got a photo of Price, fantastic, and sent it out to various people. And then we got an email back saying, that's actually Mad King Ludwig of Bavaria. and there are still no images of Price. So I did put that in. I put that as a lesson for people. Maybe. So he lived in the Swan River Colony from 1838 to 1842, collected a whole bunch of stuff, not just algae, collected a whole bunch of terrestrial plants and animals as well, and sort of sold them off or distributed them to various museums of Bavaria. He gave all of his seaweeds to a guy called Sonder, who described 80 species in this particular paper. And very annoying thing, we know where Price pretty much went, so from around Perth around to the Albany region, but we don't have specific locality information, because when he gave the specimens to Sonder just said Western Australia, and that was it. So we can't actually pinpoint where the type of localities and where these species come from, which is always a bit depressing. But these are some of the things that he will describe in that paper, this thing called Curator slendraci, which has been through multiple names because Curator changes very, very rapidly. Struvia, which is one of my favourites, and it's, you look closely at it, it's almost got this cobra-like look to it. The main branches come up, and then they produce these side branches which form a net. So you can see this beautiful intricate pattern, and this grows sort of in shallow subtidal into tidal. It's quite common. So this is a thing called Bryopsis foliosa. It's a fairly common seaweed around Point Perrin, around that area. I put this slide in, not necessarily to illustrate the seaweed. It's a green seaweed, fair enough. But what I want you to do is see if you can see an animal in there somewhere. Look just around here. To be honest, I didn't either. And I collected it because I take a photo, I have to collect things and press them and down to specimens for my photographs. And I got it back to the lab, and all of a sudden one of the branches was crawling away. And it was this thing here, which is a Saccadossan sea slug. And what these things do is they actually puncture the cell walls of the seaweed, and they engulf the chloroplasts. And so all this green here is actually chloroplast. So it's a photosynthetic animal for a very short amount of time. So it stays photosynthetic for up to about 3 weeks apparently. So yeah, so unfortunately it then went to the museum as a specimen, and we still don't know what species it is. They've been doing some DNA work on it, but it was quite an unusual find. And various other things that were described in that paper. This is actually a red seaweed, even though it looks yellow. One of the things when you talk to students, basically there's red, green and brown seaweeds and blue-green algae. And you say the green seaweeds are always green, that's fine. The brown seaweeds are mostly brown, but the red seaweeds can be almost any colour. If they've got a hint of red, then they're going to be red. But you can get yellow ones and black ones and all sorts of weird wonderful ones.
We get white.
White usually means it's dead. Yeah. Okay, so the history part of this talk is mainly about William Henry Harvey. I love the man. I think he was just a magnificent person. He was an Irish botanist. He was a workaholic. He'd done a whole lot of work on the British seaweeds. He'd done a whole lot of work on South African flora. And then he came to WA in 1854. He did a whole lot of collecting. He collected around Rottnest and Garden Island, and then had a trip down to Cape Riche. And so there's a lot of collecting done there. And he was just crazy busy. If you look at this, so this is where he collected from, or was there. And that was the purpose of his trip, to explore the natural history and to extensively collect marine algae. And he did it, certainly. So if you look at this, so he published, while he was at sea, going from WA to Victoria, he published his account of the marine botany of Western Australia at sea, 1854. 20,000 specimens during his 18 months in Australia. And he actually wrote that in one day I collected and preserved 700 specimens, some of their new kinds. I don't know if anyone's ever pressed a plant or pressed a seaweed. 700 in a day. People tell me now that he was probably a bit obsessive. Well, as a OCD, he suffered from it rather than just being incredibly busy. He suffered from depression as well. But an amazing person and a wonderful writer as well. That's one thing I find is really lacking these days in scientific writing especially. So in his book, and I will read this out to you. So, at every few yards, deep basin-like hollows of greater or lesser size break the surface of the reef and afford well-scattered, well-sheltered nooks for a variety of beautiful algae. The water in these basins is always intensely transparent, the bottom frequently of white sand, and the steep and craggy sides clothed with algae vegetation, which the brightest tints of green, purple, carmine, and olive, and the most graceful waving forms are mingled with rich variety. And it's a wonderful style. He was an incredible letter writer as well. There are books compiling all of the letters he wrote to people. So wonderful man. Some of the things that he wrote, or some of the things that he described, Asparagus armato, having 2 or 3 naked branchlets armed with reflex prickles. The frond is twice as thick as a hog's bristle. Now, I have mentioned this in talks before. I say, anyone know how thick a hog's bristle is? Nobody knows. So I must admit, I took it upon myself to examine how thick a hog's bristle is. Don't tell my boss that I did this in my work time, but I discovered a whole lot of things about how these historical units of measure actually came about. Yeah.
So you use things like that to describe.
Oh yeah. Well, this whole idea that what the inch was. It was originally the width of the human thumb, and then King Edward II of England in 1324 said that it has to be 3 grains of barley, dry and round, placed end to end, lengthwise. So that's how an inch came about. So there's all these other ones. So we ended up, a lot of the early units measure with bird feathers, bird quills and things like that. So I actually wrote to a guy, an ornithologist overseas. He said, "Can you help me?" And he pointed me to this, it was the feather index or something, where you can actually get measurements of all this sort of thing. So it was a bit of an aside, to be honest. Just for fun. All right, Asparagopsis. Some of you probably heard about Asparagopsis. So Asparagopsis it's been in the news a lot. I remember before the last election, there were the last one, maybe the one before that. The Liberal government had ads saying how seaweed is going to be fed to cows to reduce their methane output. And it's all based on this study, this idea that if you feed Asparagopsis to cattle at a fairly low rate, you reduce their methane output and therefore reduce the impact of climate change, ameliorate climate change. In theory, it sounds great. In practice, it's got to be a tough call because you have to get it up this stuff, you have to grow it, you have to get it to the cows, you have to get the cows to eat it, and lots of different conditions. People have done studies where it actually does. It's not that beneficial to the cow. But I won't go into that too deeply because this is being recorded and some people might soon. Now, I mentioned I've spent some time in Hawaii. The weird thing about Asparagopsis, it's actually the favoured seaweed, the favoured lemur of the Hawaiians. And it's an edible seaweed in Hawaii. There are regulations governing how much of it you can actually take because it was over-harvested. The thing grows along, creeps along the bottom, and it's got these fluffy tops. And so they're only allowed to take the fluffy tops and not the creeping bottom. And then what they do is they soak it overnight in water and then roll it up into balls and salt it, and then use tiny amounts of flavoured stews. So I imagine that a lot of the more active compounds are either leached out or you're just using so little of it that you don't really notice. But if you have a look here, this is actually an assessment of what's found in Asparagopsis. So, an edible alga in Hawaii composed of mainly bromine and iodine containing haloforms and smaller amounts of halogenated methanes and several halogenated ethanes, ethanols, formaldehyde, acetaldehyde, acetones, 2-propanols, 2-acetic, I can't read that one, proteins, epoxypropanes, ethylene, and butanones. So there's a fair old mixture of things that you find in Asparagopsis. So, yes, I'll leave that one. In that same paper, this was well before that, just the whole idea of feeding it to cows. It's a favoured edible seaweed. Would suggest that it's poisonous to eat, but nobody's ever got sick, probably because they only tiny amounts of things. Okay, so Asparagopsis. So some of the other things that Harvey described, this thing called, it's now known as Dictyopus australis, beautiful species marked with dotted lines like a leopard skin. Clerpa geminata, one of the unusual clerpas. Called Cloyodema. A lot of the genus names have been changed over the years because of the way taxonomy works. Things get split apart and moved around. Harvey had a pretty good set of most of the earlier collections, but he was missing some of them, so he couldn't. There were some things that he described that had actually been described earlier. I think called Cloyodema. Martensia. This has got to be one of the weirdest seaweeds. So what it is, it's basically a flat sheet, and you can see that flat sheet part there. And then every now and again, what it will do is all of the cells at the margin of the flat sheet will turn sideways and grow as a net. And you can see this sort of net-like thing here. These balls here, they're actually reproductive structures on the surface of it there. Red seaweeds have got probably the most complex life history of any plant, to be honest. Actually have 3 different phases that cycle through. This is what that looks like. So this is the sort of the flat part, and then all of a sudden the cells turn and make this beautiful net. Why they do it? I don't know, but it's nicely photogenic, so it might be a picture of it. A few of the other things, there's Calamenia cribrosa, we put into a Layamenia a few years ago. This is the Griffithia that I mentioned before, a string of ruby-coloured beads. Curator australis. one of the things that did happen with this one, this sort of reddish patch on there, Harvey actually thought that was a separate thing growing on the surface of something else. And so he described it as a parasite on a thing called Anthera, which it wasn't. It turned out it was a genus on its own, a thing called Rhodopeltis. And then others that are sort of iridescent. This is a sort of blue, iridescent bluish underwater, but as soon as you take it out, it's a red colour and interesting. This is an odd one. So this thing called Penicillus nodulosus. Harvey recorded it all over at Rottnest Island. So it was common at Rottnest on the sand-covered reefs of Rottnest. So this was in 1854. I've never seen it at Rottnest, and the furthest south I've seen it is Cervantes, so about 200 kilometres north of Rottnest. So when Harvey was there, maybe it was a very warm Northern current for that year, what we used preceding it, because it's very much a warm water species, but doesn't seem to occur at Rottnest anymore. So it's a very, very interesting observation. Shameless plug. All of these photos are in. Sorry, that's good. Okay, 640 species. Alright, so when Harvey went back to Dublin with his huge collection, he then described, he put out 5 volumes of a thing called Phycologist Australica, History of Australian Seaweeds. So there's algae from WA, South Australia, Victoria and Tasmania, and a synopsis of all known seaweeds. One thing I would always love to point out about this thing here is how he writes his qualifications. So he says, a member of the Royal Irish Academy, fellow of the Linnean Society, and a few other things in there. And I love this one because I would love it as an academic. I was an academic at one stage, so as an academic, be able to put at the end of it, etc., etc., etc. So he was a well-read man and professor of botany. And he also, so every one of these, the seaweeds, the 300 species he included in Phycologist Australica, were illustrated by these colour plates. Very, very intricate watercolours, and he did these as well. He did lithographs, so he did the actual, the basics of it, and then apparently all the watercolour, the colouring in part was done by a team of ladies, apparently. So that's all I've been told. And these are some of the things. So this is a specimen from Point Perrin. This is Harvey's description of it, and they're still very, very applicable and usable these days. One of the things he included was, at the time, was called Eucuma speciosum, which is the jellyweed of the early swander of the colonists. So you can find a lot of it around the Perth area. It washed up after storms. Very, very cartilaginous thing, and what they used to do is boil it up and use it to make jellies out of it in what was one of the colony. Now known as beta ficus. Okay, so Harvey, as I said, unfortunately, he was only in his 40s when he passed away, and as I said, he was an amazing character. He the Christmas tree of Noxia. So in his letters, he actually recognised that it was probably a parasite, just by observing that the way it was growing. That was a hemiparasite. He wrote this in his letters, and that was years before it was actually proven. So quite amazing. So following Harvey, not much happened. There's not a lot of work being done on seaweeds. A few people were, until this gentleman came along, Brian Walmsley, from the University of Adelaide, and then later at the herbarium there. And Brian he was an ecologist to start off with, but then he realised that he couldn't actually put names on a lot of things he was looking at, and so he then became a taxonomist. And so he eventually wrote these 6 volumes, one about the greens and the seaweed, green algae and the sea grasses, one about the brown algae, and then 4 volumes of red algae, which is people ask you, the sort of relative diversity of the different groups. Greens actually shares a volume with the sea grasses. Browns has one of its own, but then there are 4 volumes of red algae, an incredibly diverse group. And can be quite difficult to work on, but that's part of the fun, I suppose. Sadly, he passed away in 2011. So one of the things so when Brian was working, and Brian was actually my supervisor, so I count him as my academic grandfather, I suppose. there was nothing, no such thing as DNA sequencing. Obviously that only came in fairly recently. And so a lot of times things that were attributed to or thought to be the same as species occurring in other countries. For good reason, they look exactly the same. This genus called Halimeda, which is a calcified grain. It doesn't look like it's calcified, but it actually is. And it's it's usually a tropical genus. There's multiple species up north, but then there are a few of them there's two species around the south coast, also around the southwest coast. This one's the most common. And Wolseley named it, so Harvey collected some, okay, and he distributed a thing called Halimeda macroloba. But Wolseley put it in a thing called Halimeda cuneata, which is a South African species. Right? So, is it actually the same as the South African species? And in a sense, it's a good call. It looks exactly the same. You look at the structure of it, it's all the same. There are odd things in that they are all just one cell. That whole plant there is one cell, even though it looks like it's multiple there. It's actually essentially a filament that hasn't got plugs in it. You can grow these things in your fish tank, and if you go out at night time, they're white because a lot of the chloroplasts actually migrate internally in the dark and then come back when the light comes on. So we did some DNA on this thing, and what we found, so this is the Halimeda cuneata from Australia, and this is Halimeda cuneata from Africa. Some of the specimens are called Halimeda cuneata there. Oddly enough, there's another one as well. And so there's two species that could be cuneata in Africa, but neither of them are the Australian one. And this is I suppose, the beauty of voucher specimens and keeping collections. That Agard, Harvey sent one of his specimens to a guy called Agard, and he actually described, he named the species based on that Harvey specimen. So we know that it came from southwest Western Australia, and it's not a South African thing. So he described a thing called Halimeda versatilis. And we were like, well, okay, how do we know where Halimeda versatilis came from? We know it's from the southwest. But Harvey put this thing called C here, okay? This is the number that he put on the specimen. And if you actually look in his code, C is Cape Ridge. So we know that the specimen came from Cape Ridge. It's definitely a Western Australian thing. And so we adopted the name versatilis. That was a good example of it. Sometimes things don't work anywhere near as conveniently as that. So we put a new name on. We have the Western Australian Halimeda. Last few years, I've been getting very interested in Harvey's species. So a lot of them, you can imagine, this is hundreds of years ago. A lot of them have been restudied. But there are a whole bunch of them, what I call fuzzy reds, that he just called a thing called Calathanion and Coronospora, but nobody has actually known what they are because you need reproduction and you need much of further study, and nobody's ever looked at them. So I thought maybe I'll try it because most of them were collected from WA. So I went out and did my best to try and recreate the collections that Harvey did, and managed to get most of them. This is the thing that he called Coronospora gracilis, and we got some. He collected it from Garden Islands, and we collected some from Cape Herring, so very, very close. This is one of Harvey's original specimens. This is the stuff growing on Sargassum down Point Perrin. This is what it looks like. We never found actual reproduction, but what we did find is these things here. Which you can see these grains here are starch grains, and so it's actually a vegetative reproductive structure. So it'll fall off and reproduce the parent plant. So it's a propagule. And so not sexually reproductive, very, very distinctive, and they actually matched a thing that I described a few years back from the Abrolhos called Gyriella, where we did have reproduction. So it sort of shifted the name over. And then another one, similar sort of thing, and the same thing as Coronospora australis. We collected some from Walpole and Norway, and we actually managed to get reproductive structures. And so, based on all of this and the DNA, it's now put into a genus called Desica shariella. So a lot of these Harvey species are now hopefully being put into their correct placement. Okay, so that's a bit about the southwest or the Perth region. What about north? When I first came to WA in '85, I I thought, oh, what's known about the northwest? And so I did a bit of a literature survey trying to find out what was there. And when you look at it, in several thousands of kilometres, there are only 28 species recorded, algae recorded in literature. And half of those were things that were associated with mangroves. And so there was a group that went out and said, we're interested in mangrove epiphytes, and they collected a lot of those. But nothing was really known about any of the northwest flora. So I was very, very lucky that the museum then had lots of expeditions up into the northwest, and I was invited as sort of the token botanist to come along, which was wonderful. Some of the things why the northwest is not particularly the best place to go collecting seaweeds or any sort of marine stuff. Tide range is one of them. So it's the huge tide ranges up in the northwest, of course, which is good and bad. I mean, the bad side of it is that all that water moving around at a huge rate makes for lots of currents and eddies and murky water and all sorts of things. The good side of it is that you go there at the right time. What's normally subtidal is actually exposed, and you can go and collect things, just walk along the reef. But so it's not the best place, and of course it is remote. Just to get there is difficult. And of course the other things. I have to say this is not a stock photo. I've made field trips for multiple years to various places. We started off in the Dandara Archipelago and then moved up further north and ended up around Ashmore and Hibernia Reef, places like that. And one year I couldn't go, and I had a PhD student, and so I thought, great opportunity, Grando, you can go on one of these trips. They were fully geared up, ready to jump into the water when this thing popped up next to the boat. And so it still fills me with dread every time I think about it. The split second, it's sort of like one of those things where you're almost prepared to put yourself in certain situations, but putting somebody else in it. And of course sharks, which is probably of the two, I think I would prefer to be a shark than a crocodile, but obviously don't want to be either. This one's a bit of a furphy as a photo, because this is actually a place called Asho's Gap in Coral Bay. You've ever been there? It's a wonderful place to go for a snorkel, and you can see here you can see actually four, you can see up to about a dozen sharks at any one time. And it's a cleaning station. So it's a particular reef where the sharks come in, and they come in and then they'll just sort of go semi-45 degrees, open their mouths, and the little cleaner rats will get in there and take out all the little bits that are stuck in their teeth. Quite impressive. And then the shark will just swim off. So they're not there to eat. It's quite an amazing thing to see. Yeah. But they do bite. This happened to be a trip I did up to the Rolly Shoals with a lady from Ames in Townsville called Katerina Fabricius. She was a soft coral expert, and she was my guide buddy. And she was very, very matter-of-fact German lady, and we were sort of pottering around on the surface for a little while, untangling a rope. And she called me over and she said, "I have been bitten by a shark." So what does that mean? Do we keep going? And then she showed me her leg, and no, we didn't keep going. So she actually had a shorty wetsuit on, so about this point here, the wetsuit protected. You still see the punctures here, but they're quite deep. She got back on board, and we actually had a couple of shark biologists who informed us that sharks have got very, very clean teeth. And so they just irrigated it with saline for a day, patched her up, and then the next day, or 2 days later, she was back in the water. A little bit wary, let me say that. The very next dive, a hammerhead swam right underneath the two of us. Oh, look at that. But yeah, she was feeling a little concerned most of the time. She actually, she's got this wonderful set of photographs where she was, a turtle came up to her, and she thought, oh, this is great, take a nice close-up photo of the turtle, and then the turtle got closer and closer and closer, and tried to nibble at it, and she ended up she had a slate that she was writing notes on. She shoved the slate in the turtle's mouth, so it didn't bite it. So yeah, so I don't think she's ever come back to WA. Maybe the Barrier Reef's safer. Other things that you find up there, sea snakes. This one looks particularly ominous because of the way it's lit. It's just lit from above. But yeah, sea snakes all over the place. And most of the time, they just come up, they're curious. People tell me they're short-sighted, so they'll come and check you out. It's always a bit disconcerting when the sea snake's making a beeline for you. They'll just come and check you out. The they're pretty well harmless, but then people will tell you stories about, you know, them getting caught in nets and stuff like that. If they're hassled, if they're, you know, something annoys them, then of course they can react. But diving with them, not so bad. Some of the northwest is a place called Hibernia Reef, which is just north of Ashmore Reef. Beautiful coral reef, clear water, beautiful fish everywhere. And then sometimes it looks like this. And so we, the practice was to lay down a rope and follow the rope and then come back to where the rope went back up to the surface again. And seriously, there were quite a few moments where you were very happy to have that rope. Especially when you hear the tap, tap, tap on the tender and then you come to the surface and go, the crocodile's been sighted in the area. Thank you. So yes, we weren't being crazy, but I think we were just off being crazy. Yeah. Okay, so some of the things that we got from the northwest. This is actually from James Price Point, his title thing. And you look at this, I think most people look at this and go, it's a sponge. And I probably would too. But actually you can look at the tip here, and there are certain red algae that have a symbiotic relationship with the sponge. You never find that alga, the seaweed, growing independent of the sponge. And there's some around the coast here as well. And so the alga is on the inside and it has a sponge coating. And this one was actually described by a lady called Anna Weber van Bos. There was a Dutch phycologist being people that study seaweeds. A Dutch phycologist, and she managed to get on board the Savoga expedition, which was a very famous exploration around 1899, 1900. And the Dutch now, a year on board this thing. She managed to get along because, well, normally women wouldn't be invited on those sorts of things because just the times that they were. But her husband was the professor of zoology at the university there, and so she got a berth, which was wonderful. Actually follows where they went. Now, I'm not sure where it starts, where it ends, but it gives you an idea of how far they went in their year. And she ended up writing a book called Een jaar upon the Savoga, which I'd love to translate into Dutch, but my Dutch isn't that great. And then it's in translation of it. But yeah, so she wrote this book, and then she wrote a whole lot about a whole lot of studies, and she was actually the first. Oh, sorry, I'll keep going. So this is Anna Weber van Bos and Wax, her husband, so 1890. This is her on board the Savoga. So here she is with her cat. She's with her cat, and all the crew. So this is the scientists, and then the crew in the background. I don't think the cat actually made it home. So she took a monkey home with her. So different times. But I love this photo. This is, collecting in the tropics. So, long skirt, the puffy sleeves, and the cat sort of docked slightly to the side, collecting seaweed. So, yeah, quite amazing. So when she went back to Holland, and she described all the results, and she was the first Dutch lady to receive a PhD. It's quite impressive. This is one of the things she collected, actually. It doesn't look like much, but it is actually these are the original specimens of a thing she called Phanerozoa in the Western Herbarium. And this is her drawing, so this is the inside of the seaweed, and they have these really weird sort of filaments coming out, and then these odd green cells floating around in it as well. This is something we collected from Shark Bay, which turned out to be the same thing. And we had the same sort of arrangement, so this is the seaweed part, these filaments here, and then this is all sponge spicules. And then some odd green cells growing in. So it's a symbiotic thing again, where you've got the alga and the sponge, and in fact a unicellular green growing amongst it. So how do these things actually reproduce? And so what we found is, so this is the alga with the sponge coating. When it becomes reproductive, it just produces these little sort of bulbous leaflets that have all the spores in them, and then they release the spores. Big question is, how do those spores then catch up with the sponge and recreate the plant in the first place? And because you're going to do so much effort here to actually get the spores away from the sponge. But so we did some DNA on it, and again, same sort of thing. She put it in a thing called phycologist, but actual phycologist is down here somewhere, and this is what we had. And so it was actually a new genus called Spongiflea, which means this is spongy bark. So these are the places that I've been up north. Wonderful trips. Unfortunately, we don't get to go anymore because they sort of feel like they've done their dash and that sort of thing. These are some of the new species, a thing called Ganoderma robustum. It's a calcified seaweed. Weird, wonderful thing called Gibbsmithia. Sort of gelatinous beak, actually has a firm stalk. And so we had 523 species of algae, so it's a huge increase. 6 new genera described, and a whole stack of new species. And so several PhD projects, and 2 books ended up, we ended up producing 2 books on this flora. I put this one in, this thing called Prodia. I called it Prodia Sophie. My daughter's name is Sophie. Again, it doesn't look like much, but it's a red alga which is actually filamentous. And when I said before about red algae having incredibly complex life cycle, this is actually the egg cell of the female, and it produces, you can't see it well here, but it produces a hair, receptive hair. The red seaweeds don't have any motile cells. Every other seaweed has a swimming stage that can go from one to the other. The reds don't. And so they have this receptive hair. If you look closely into these tiny cells on the tips here, they're the males, and then the males are actually released, and then somehow they find their way into this tiny little hair and fertilise it, or they deposit their nucleus, and that goes down to this egg cell, and then you get the join, so the fertilisation, and then you get this separate generation growing on the female plant. So it's a diploid generation, so it's the product of fertilisation growing on the female plant, and it's classed as a separate generation. And so it turned out to be a new species in this Prodia. I did call it Prodia Sophie. This was my daughter at the time, who's a figure skater, and I figured, I just love the colour and the flowing and everything. But I didn't tell her at the stage. I said, you're probably going to be only 14-year-old, or something like 13-year-old in WA that has a seaweed name for it. By the time it eventually came out, she was probably only 18-year-old, because it took a while to get the book out, but it eventually came out. So these are the 2 books that came out. It was quite nice. All right, if I've got time, have I got time? Yeah. All right, so I thought, whenever I've given these talks before, people say, what can you eat? So I'm trying to put in a few examples of things that you might forage on the shoreline that are essentially equivalent to some of the things. Seaweeds, all sorts of things go in seaweeds. Obviously, you can pay models to do pretty much anything, including breaking yourself with seaweed. Used in a lot of in cosmetics. It's very, very hydrophilic, so it's good moisturiser, a lot of seaweed compounds. I'll read this one out. Seaweeds and other algae, as true for many plants, can be weedy pests, toxic, food, a source of various compounds, useful in the garden as a fertiliser, useful in environmental remediation, implicated in environmental degradation, decorative, inspiration for architecture and artistic design, used in medicine, used in assisting birth, if anybody wants to know that one, used in dentistry, used in laboratory, in cosmetics, as a food additive, a source of vitamins, colouring agents, in submarines, and potentially a source of fuel to keep our cars and planes moving. So if anybody asks you what day seaweeds have, there you go. And the assisting birth is actually there's an old Chinese practice, apparently. And what they would do, they would get the stipes, so the big stems of kelps, when they were dried out, and when they were hoping to extend for pregnant women hoping to dilate the cervix, they would place these things into the appropriate place. I won't go into too much detail. And by absorbing the moisture, it would gently expand and dilate the cervix that way. So I don't think it's used very much these days, but there you go. All right, so there's below and food. Obviously, this is the good stuff, vegetables from the sea. This is what most people would know, of course. Nori. So it's the black stuff that wraps around your sushi rolls, which is of course seaweed. It's a huge industry, and initially nori was reserved for the very rich and I suppose kings and whatever. I can't recall what category they were in, but because it was sort of an accidental fit, you hope it would grow and you collect it, but they worked out that you can farm it, and this is sort of a farm in Japan, of course. It was actually the big change was from a phycologist, a lady called Kathleen True Baker. She worked out the life history of this thing, and so rather than it being just a random thing where something settled, they worked out that the alternate stage of this red seaweed actually had a microscopic stage which you could seed onto ropes. And so they seeded all these things onto ropes, they put the ropes out and they grow back up grow into the plants. And because of that, she's probably the only phycologist with a statue for it. She calls the Mother of the Sea, and April the 14th of every year they have a special day commemorating her service. This is our porphyra. If anybody goes down the beach, probably in winter, and you see the green alga, but you'll see these sort of strange purpley ones. It's a porphyra, which is an edible one. Probably a little bit tough compared to the others. So, yes, I spent some time in Hawaii, and they do actually have a thing called a spam masugi. So you get the spam, the rice gluten, right, and wrap the seaweed around it. And they've got the special spam, which is 25% less sodium, but probably still a lot of salt. Spam masugi. I tried once. But yeah, seaweeds and all sorts of things these days, lots of snacks. I don't know why you bothered making a dress out of this stuff, because I'm sure it probably wouldn't stand up too well from the first rain. Alright, so some of the bad ones, or some good and bad. So this thing called Undertakes pinnatifida, wakame. And again, if you've eaten in Japanese restaurants, you've probably eaten some of this stuff. Native to north coast of Japan, Korea, and China. Has been introduced to all sorts of places. It's an introduced weed. It's quite common in Victoria and Tasmania now. Nobody really knows how it got there, probably hitchhiking on the bottom of ships. But yeah, it's been in New Zealand, got introduced in California as well, all sorts of places. So one of the things with seaweeds is if they get in, if you've got an invasive seaweed or a pest seaweed, it's almost impossible to get rid of them because they don't have vascular tissue, so you can't use a fertilizer, you can't use a herbicide on them because they don't work. The only thing you can do is essentially just nuking the whole thing, which is what they did when I have done in a few places. You kill everything in the hope that what comes back is going to be native. What they're doing in New Zealand, they're actually harvesting this stuff, selling it for food. So, good idea. So some of the other things, this is macrocystis, which again we don't have here, but we do have in Tasmania. And I won't go through all this, but it's used for all sorts of different things. And it was commercially harvested for quite a long time. They used to have these macrocystis ships, and essentially they would just cruise along the kelp beds and take out the top couple of litres of macrocystis, and it was used for alginates. The stuff grows so fast that they probably wouldn't even notice. Unfortunately, you wouldn't want to do this certainly wouldn't want to do it in Tasmania because the kelp forests there are so small and they're being pretty well decimated now anyway by climate change and urchins. So yes. The Villia I spoke about before, which was used as food and medicines as the water container. This week, if anybody's been up to sort of Philippines and Bali, places like that, in Indonesia, you might have seen seaweed farms up there. They're growing a thing called ficus, which is not eaten, but it's harvested for carrageenan, which is used as a thickening agent, so it ends up in sort of toothpastes and all that sort of stuff. A lot of it ends up in pet food, I think. A huge industry, very, very labour intensive. So this is all the ficus here, and they essentially just harvest it and then they tie little bits onto the ropes again and put it all out into the thing. It's it's a tough life, and it probably doesn't give you much money because most of this stuff is bought by large multinationals that don't give the actual farmers the money that they should get. But in terms of it's not particularly harmful to the environment, other than perhaps shading out some things. But it's quite well done. Anybody, there was a film not that long ago, very, very trashy one where some royalty in Indonesia was a seaweed farmer, and apparently he was earning millions out of this, but I don't think it was truthful. I mentioned this one before, which is the same sort of thing. I did mention Asparagopsis. This is the taxophorus, which is the other species. which is the renal kohu, and I mentioned here again, it's added to stews and used in small quantities because the flavour is penetrating. So what can I eat? So look, I think mostly edible, but I would be a bit wary. I would certainly eat any blue-green algae, it's cyanobacteria. I know some freshwater cyanobacteria are sold as health food products, but I think the marine ones are probably less so. Some clervicin have toxic compounds, so they have a thing called clervicin. So there are some clervicin. If you are in the tropics, there's a particular clervicin very often used in salad, but the local ones, or some of the others, can be quite dangerous as well. So certainly, if you're eating anything and it's got a very bitter taste to it, I wouldn't continue. So some of the suggestions, Gracilaria, Dictyopteris, all of which you can find pictures of somewhere, and possible cooking. Gracilaria, the Hawaiians do a wonderful one where they take the Gracilaria and they mix it with raw fish and a bit of sea salt, and it's very nice. The Gracilaria actually has a nice crunch to it, poke bowl they call it. That's Gracilaria. There's Gracilaria here again. So I realised I'm taking way too much time. Dictyopteris, again, it's a Hawaiian one. That one I think is a bit of an acquired taste, let me say that. They let it ferment a little bit in a bucket first. And ulvas. And playful ulva. So I'll whip through these, but these are, I'll leave the presentation with the crowd, anybody wants to look through it. Because I realise we're running out of time. First Theka, and again the clervis. Clerval is the one that actually is used as a salad vegetable. Yes. Avoid the cyanobacteria. Legalities. I actually asked somebody asked me, can I collect seaweeds? What's the legality of it? And it's not as simple as you might think. Depending on where it is, who has governance over the area. Certainly, if it's a marine reserve, you need a permit to collect seaweeds because they're counted as flora. The fisheries department always just used to define seaweed as fish, and so they were classed under that sort of thing. But there's a whole lot of things, even clearing, Environment Protection Act. So there's a whole lot of different things, but wading through all of that, my feeling is that if you take parts of indigenous seaweed completely so much on my scale, other than in marine reserves, you don't require a government authority. What I wouldn't do is rip it off the rocks, so I don't do anything else as harvest and then from the top and let it regrow. I'll finish with, or nearly finish with this one. This one appeared in my letterbox quite some time ago. No, it appeals to me. So ficus is a brown seaweed. We don't get it here anyway. It contains ficus, totally natural ingredient, using weight loss somehow. And apparently what you do with ficus is you wrap it around your problem area and you can lose so much weight. But it's when you think more kilos fat. And the best thing is, and yet you still eat 5 million. So I wouldn't believe everything you read.
No.
Now, people often ask me why I like being a taxonomist. There's certainly a lot of this discovery. I can get to go and take pictures and do all that sort of stuff, and you get to name things. But there's also the connection with history. Anna Boss was a Dutch lady. I could actually see her specimens. I could get her specimens, and so things that she'd actually touched and use it. We had a visitor at the herbarium once whose great grandfather was a botanical artist, and she said, "Oh, would you have any of the specimens that he painted from?" And I said, "Well, we'll find them. We'll find some." So she came back with a friend of hers, and they must have been pretty scrappy things. But she was almost in tears because it was this contact with her dead relative, her dead ancestor. The lady who was with her was like, But anyway, years ago when I was at Melbourne Uni, doing my PhD, when Barry Humphries was obviously still alive, he asked the professor there for pictures of the naughty bits of gladiolas, because he wanted to put them in the program he was giving out to people. Barry Humphries being Barry Humphries, throwing out gladies. his name Edna. Anyway, so to return the favour, he said, I'll come and give a talk in the lecture theatre, which he did. He was asked one question and he spoke for an hour. It was amazing person. But he told this story, which I just love, which sort of reflects a similar sort of thing. That, you know, when he was young, he shook hands with Percy Granger. He would shake hands with Delius, he would shake hands with Grieg, he would shake hands with Liszt, he would shake hands with Beethoven, he would shake hands with Haydn, and he would shake hands with Mozart. So he could actually claim a full connection all the way back to Mozart. And my biggest regret is that I never went and shook his hand. but I told this story at a student camp once, and one of the students there, her family was family friends with Barry Humphries, and she had shaken his hand, so everybody went up and shook hands. And with that, I'm sorry I went a bit over time, but happy to answer any questions. Thank you.